Okay, thank you all for coming to this wonderful session this morning. We have two amazing speakers. This presentation is going to be about funding the future, why ending animal testing is a long-termist prerogative. And it's presented by Dr. Brinda Poojary, who's been working in the field of non-animal methodologies alongside academic, industry, and policy stakeholders, and has worked to bring about important regulatory shifts towards non-animal testing in India. And Sreyoshee Sengupta, who's a PhD student in the field of developing non-animal models at the Technical University of Denmark and the National Research Center for the Working Environments in Denmark. Please welcome our two speakers. Thank you very much.
Can you hear me? Great, perfect. So I have a question for you. When I say animal testing, what area or field of study where animals are used comes to your mind? Sorry? Yes? Any more? Exactly. That's perfect. I think both your answers sums up animal testing that broadly comes under two umbrella terms. One is in the field of biomedical research, and the second one is in the field of regulatory testing. Thank you. So, biomedical research includes basic research, so basically understanding the basic biological processes in the human body. It also includes understanding onset and progression of diseases. To do such experiments, researchers are required to justify animal welfare committees as to why are they performing these animals. And this justification includes giving a detailed information from purchasing the animals, the suppliers that they get the animals from, transport of animals, maintaining the animals, which includes the number of animals that you would house per cage, the frequency and the interval of food that you would supply to the animals, and the environment which they are living in. So you need to give such detailed explanations. The issue over here is that there is no limit on the number of animals that you can test using these in the field of basic research. Another area is in education and training. All the researchers are required to be certified to handle animals or even plan animal testing. And this includes by attending courses that are legally certified in the respective countries. And in most of the courses, the surplus animals that were used in the basic research are used in these courses, but again, there are no strict regulations on the number of animals that could be used. It's the same in the discovery and development of drugs, vaccines, and medical devices. I have another question for you, or more an activity for you. What do you think comes under regulatory testing, right? So now look around the room. Look at the clothes that you're wearing, the seat that you're sitting on, the soaps and shampoos that you've used this morning. All of these need to be tested for their safety, both on humans and on animals. So this includes safety testing of drugs, of vaccines, and of the consumer products like soaps and shampoos, also including cosmetics. And this is also these safety assessment testings are also important to assess environmental safety. The issue again here is that even though you relatively have better regulatory guidelines, which states the minimum number of animals a researcher is supposed to use, but there is no cap on the maximum number. So if the regulation says 10 animals, the researcher has to use 10 animals, but he or she is also allowed to go more than 10 animals. I'm sorry. So again, another question. Can you guess the different types of animals used in animal experimentations? Sorry? Yes, and? Mm-hmm. Yeah. Yes, very, very good. Yeah? So, all your answers, or most of your answers are over here. I'll give you like a second or two just to see the number of animals that are used in animal experimentations. Now, the issue is that there is a transparency gap. The first transparency gap is lack of reporting. Most of the countries, they do not report the number of animals used per year in their countries. The issue with such lack of transparency is that we do not have, it hampers the efforts to have a more standardized and harmonized system of conducting animal experiments, or even understanding the number of animals used. And there are just 5 countries, or including the EU member states, or all the countries in the EU member states that report animal testing. But there are certain discrepancies here which I would talk to you about in the following slides. The next transparency gap is the species and reporting discrepancies. First thing, definition of animals. There is inconsistency in species recognition. So the issue is that even though most of the countries that I had listed above, they do report, US, for instance, they do not consider animals like mice, amphibians, reptiles as animals. And that also includes invertebrates. That's most of the countries do not regard them as animals. And the second part is you do not, the number of animals used just to get their tissues are not reported, which also includes the surplus animals that I'd spoken about that are used in different animal experiments in basic research. Sometimes it could also include regulatory testing. And lastly, it's the animals that are used to maintain breeding of animals. Something that's a bit jarring is that animals that have any genetic defects are also maintained, but are not included because they would like to have more offsprings, hopefully with the same genetic defect. And the third transparency gap that I would like to discuss about is the intensity and scale of pain in animal testing. So an example would be understanding the potency of rabies vaccine. So for a vaccine to be marketed, it is essential to test them in animals. And the way the testing happens is that you have 2 batches of animals, one batch called the control, which is infected with the virus, and the second batch, which has the virus and the vaccine that they are trying to market. The first batch, the control, they are killed as a part of, to enable less suffering to animals the moment they start seeing symptoms of viral infection, which in a way is a good refinement strategy, I would say, but things have to be changed. The second batch of animals, which is the test animals, are only killed after they have seen if the vaccine has worked on them or not. Then, now that I've given you a brief of the animals used, what kind of transparency gaps we have, would you like to guess the number of animals used per year, which includes both biomedical research and regulatory testing? Just a ballpark would be fine. Sorry? Close? So, in 2015, there was a study conducted which said that there were 192.1 million animals used worldwide per year. The issue over here is that this study was conducted in 2015, and we are in 2024 now, and we still do not have a detailed statistics of the number of animals being used. The study was previously conducted in 2005, and they had seen that there was a decrease, a 2-fold decrease, I would say, in the number of animals used, and they predicted that it would decrease further, but we have no studies to prove that. So why do these tests exist? An ideal study to understand human biology would be recruiting human subjects, but it is not practical and lately considered unethical because you would also have to represent the diversity of us humans. And that was one of the reasons why animals we turned to animals, or researchers turned to animals to understand, to model us. And this is just a slide to show that we can still live in harmony with all the species sharing the same environment. So, what can we do? What is the limitations of animal testing, even though, although we knew that there weren't any other model organisms other than animals to represent us? The first limitation is resource intensive. So I think it was in 2021 that a researcher named Thomas Hartung from the Johns Hopkins University had found out that $14 billion are spent in animal experimentation worldwide for hundreds of animal experiments that are conducted. So it's very resource intensive and time consuming. The second bit that we have is challenges with translatability. What do we mean by that? So most of the cancer research that has been done in mice. A famous scientist called Richard, I don't know if you can read the name over here. His name is Richard Klausner from the National Cancer Institute in the US, has stated that the history of cancer research has been a history of curing cancer in the mouse and not in humans. Yes, we do understand the disease much better, but do we have a cure? No. And the researcher that I just spoke about, Thomas Hartung from the Johns Hopkins University, has very aptly stated that humans are not 70-kilogram rats. We live longer, which means that the drugs that are, or the diseases that we encounter have a different onset and different progression. And it also means that we have the ability to evolutionarily adapt to these diseases. Another limitation is the global harmonization. The issue with, I think I'm running out of time, but I would just like to say that in the field of global harmonization, we do not have standardized animal testing models. So example, the vaccine examples that I had just given. So it's very difficult to have procedures that all countries can follow. And like I said, there's an example of the target animal batch safety testing that was recently stopped in India, and it has been banned in the EU for a while now. And the issue over here is that these target animal batch safety testing involves animals which are produced in India, or which was required in India. They would still need to do these batch testing to sell them in India, but remove the batch testing when they would have to import it to the EU, which means it's very resource intensive and time consuming. And now I would have Brinda, who would give you the way forward from all these issues. Thank you.
Thanks, Sreyoshee, for all those insights. And I would just actually, because she didn't have time, I would like to go to the previous slide and talk about the... Thank you so much. I would like to talk about the target animal batch safety test that she mentioned and why lack of harmonization is something that is extremely crucial. One is because target animal batch safety test, just to give you an example, is one of the regulatory tests that is extremely essential and not extremely essential, it's mandated by different regulatory bodies. And in India, it used to be mandated by the Indian Pharmacopoeia Commission, but the EU had banned this test. And then it also decided to ban any vaccine that has been, ban the import of any vaccine that has been tested on animals for batch safety tests. This is just one of the plethora of regulatory testing that is done for releasing a vaccine into the market. So when industry, because India is a huge producer and exporter of vaccines, the Indian companies had a huge problem with this because they had to make separate, they had to produce a separate batch in order to market its sale in India, and they had to test that using target animal batch safety test. And also they had to have a separate batch which was not tested with the target animal batch safety test. So this not only caused a lot of logistical issue for them, but it also caused a delay in exporting the vaccines and the vaccines reaching the consumers that needed it. And also, in certain instances, it caused the vaccine to expire and a lot of wastage. So because of that, that brought about a change in the way that regulations, regulatory shifts took place in India, and very recently it has been waived off and it is not required. So I just wanted to point that out. Okay, so shifting paradigms. And now just to answer the question that all of your brain cells have, is this over yet? No, it's not. The saga of animal testing continues with me. Now, as Sreyoshee explained, she explained what does not work. So I'm going to explain what does work. In vitro models. So what are in vitro models? Can we hear this? We hear this word a lot: in vitro, in vitro, in vitro, in vitro testing. But can someone give me just one word for what in vitro model is? Even if you have 3 words, I'm not going to penalize you. So outside the body. Thank you so much, Apurva. So in simple terms, in vitro just means outside the body, and this is one technology, and now you may think that it looks very simple, right? Does anyone identify what this is? No? Okay. This looks really simple, but its job description is not. So this is called an organ on a chip, and it is used to mimic a system. So say I have a lung and I want to mimic a lung. So if I wanted to mimic a particular function of a lung, then I use an organ on a chip. So what I do is, if you can see, there are multiple, this is a really tiny silicon chip, and there are multiple channels that you can see. There's blue and then there is red. So in one of the channels, this is of course attached to a lot of other very complicated systems, and through one of them you pump air into one of the channels, and then you pump blood into one of the channels, and between the 2 channels there is a barrier made of cells which mimics the air and blood barrier inside your body. So now, if I wanted to test, say, the effect of cigarette on human lungs, then instead of using innocent animals that definitely do not have any interest in smoking cigarettes, instead of using them and forcing them in containers and making them forcefully breathe cigarette smoke, I could just use another of the very complicated system that is attached to this, and pass smoke through this, and then observe the effects that it shows on a microscopic level in the system within the body. So if you want to know, this is just the tip of the iceberg when it comes to these kind of technologies. So if you want to know more about it, you can just hit me up or Sreyoshee, and we can talk more about it. So one of the things that is being done right now is to have multiple of these chips, like a brain on a chip, skin on a chip, intestine on a chip, lung on a chip, pancreas, liver, kidney, bone, adipose tissues, etc., and then align it all to have a human on a chip. So that is something that is being worked on. Next is in silico methods. So in silico directly translates to the silicon of a computer chip. It refers to the silicon of a computer chip, and this is used for prediction using computational approaches. So it can predict drug toxicity and drug efficacy without any animal testing. It can also be used to identify promising drug candidates and optimize them, and then optimize their development. Big words, long sentences. Integrated approaches to testing and assessment. It looks big, but it's not that difficult to understand. So what it basically does is you can use in vitro and in silico technologies and methods and combine it in order to assess the safety of chemicals and it enables scientists to understand it better before they decide whether they need to test on animals or not test on animals. So to make it very simple and to explain it very simply, I'm going to give you an example. If you were approached by an ardent meat eater who is very addicted to meat, and they said, can you give me, I want to see what meat replacement tastes like. Are you going to give them just mushroom or raw jackfruit and tell them that this is what meat looks like, or this is what alternative to meat tastes like? You're not going to do that. You're going to give them something that is that is made using a combination of different things that mimics the taste, texture, and different components of meat. And that's what you're going to give them, so that it makes it easier for them to transition and to know that there are other alternatives to meat, and then they can have that. So you try to mimic meat as much as possible, but there is no one-to-one replacement. You need to have multiple things come together in order to have that one thing that can replace meat. So IATA is one of those things. Integrated approaches to testing assessment is one of those things which does something similar, where you take multiple of these tests and then you bring it together in order to create a whole framework that is that mimics the human biology. Because the goal here is not to find an alternative to animals. The goal here is to mimic the biological system, which is extremely complicated. So now everyone must be thinking that, oh, Brinda, there are so many cool technologies and methodologies, but then why is there still animal testing? And that's because there are a multitude of barriers to this. So the first one is regulatory barriers. So now regulatory barriers, first of all, it could be because of a few reasons. One of them is that regulators are really risk averse. So fear, obviously, we are governed by fear way more than we should be. And aversion to risk and comfort in doing the things that has usually been done for years together is something that gives more assurance, and that could be a reason why regulators are more risk averse to adopting more non-animal methodologies rather than instead of using animals. And also, because human life is given so much importance, there's a lot of legal repercussion if there's any kind of adverse outcome that takes place in humans. So that's another reason why regulators are risk averse. And that's why, because there is high success rate in non-animal methodologies, even though there is high success rate and there's a lot of data out there, it's still very difficult to convince regulators to take up these to accept these into regulatory mandates. Second is academic resistance. So again, I think I would give a vegan example. So if we already live in a very non-vegan world, right? The systems around us don't make it that easy to be vegan. So similarly, and if I already did not feel so strongly about animals, then I think it would take a lot of behavior change in me in order to push against the flow and push against the norm and to be vegan. So similarly, as ironical as it may sound, all of the research and all of the scientific progress that is out there is governed by funding. So if grants are out there which promote only animal research or mostly promote animal research, then a scientist or a researcher is going to apply for those grants because they have to further their career, and they also have to sustain in the system. So this segues really well into the funding limitations and the need for more targeted grants which don't exist. Another important barrier is misguiding the public. So how many times have we heard like really nice, tantalizing press releases? Groundbreaking nasal spray protects against all SARS-CoV-2 variants. And then we get our hopes up. Or then we read something really weird which says drinking Coke and Pepsi may increase testicle size and testosterone production, studies say. But the thing is that, very important thing that I should say is that all of these are tested in mice. And the problem with such press releases is that they don't mention this very important intersection between animal testing and human translatability, that it does not translate to humans. And they also normalize animal testing if they mention that animals are being used. So everyone thinks that all of this is happening because animals are being used, and they can't imagine something without animal testing. So just as an aside, I know you can't see it here, but if you want more of such fun examples, then there is a Twitter handle called Just Says in Mice, and he has put up a lot of these examples, and I promise you that this is not a paid promotion of the channel. Anyway, moving forward. The way forward, what's the next way forward? We need more policy change. A common idea is that if we have a lot of alternatives and if we have a lot of available data, then things will change and policy will change. But what we have noticed is that that is not the case. As I've shown you before, there are a lot of alternatives available, but then animal testing is still way more prevalent. And that's why we need a very top-down approach to the uptake of animals. So a classic example of that is the cosmetic testing ban by the European Union. The cosmetic testing ban was first proposed by the European Union in 1993. And this was even when alternatives were not properly available, but the EU proposed that we are going to ban it. So what happened was, this pushed targeted grants, so this pushed different companies like Unilever, AstraZeneca, etc., to put in a lot of money into the development of non-animal methodologies. And so even though obviously the deadline for banning animal cosmetic testing in EU got pushed to 2013 just to give the industry more time, it was still a push in the right direction, even though it took time. And similar work is being done even in areas like drug testing and vaccine testing. So that brings us to the importance of dialogue between researchers, industry stakeholders, policy stakeholders, and animal welfare organizations. So the COVID-19 pandemic shed the light on the importance of the use of non-animal methodologies and the accuracy of non-animal methodologies, and it also fostered EU-funded collaborations between various stakeholders to start projects that start combining different testing methodologies to develop more IATAs, which I mentioned before. And of course, capacity building. So it's very important to have various programs that educates early career, established scientific policy and industry stakeholders on what the current trends are. So to summarize this, I would say that the scale and intensity of animal testing is still not understood very well, and it's not recognized very well, as Sreyoshee mentioned, and that's why this is a highly neglected cause area. This is also very tractable and a solvable issue, but when anybody thinks of non-animal methodologies, and there's a very different term in the scientific community for that, it's called new approach methodologies. And when anyone thinks of that, they only think of better systems or better testing methods. They don't really make the connection that this is something that is animal-free or without animals. So even though the end goal that we are talking about lies at the intersection of ending animal testing and human health, because there is this whole gap in understanding the connection, it takes stakeholders to work together. It leads to stakeholders working in silos, because of which it pushes the timeline even further away of the complete replacement of animal testing. And also we need to have a top-down approach, as I mentioned, for this end goal, so that there is more funding that goes into the profit and non-profit sector that works in the non-animal methodology area, and also have policy changes top-down, so that there is more incentive for the development and acceptance of non-animal methods. So just to end my talk, to address all of these gaps that we have mentioned, Sreyoshee and I are working on a multi-stakeholder approach to make these opportunities in the field more accessible. So in case you would like to get in touch with us to know more, or you think that we should reach out to someone who's working in this area and who you think would be a great resource, please feel free to just scan the QR code and leave your contact details there. And thank you so much for being such a great audience.
Awesome. Thank you so much. We're going to move the chairs to the middle. Meanwhile, we're going to take questions on Whova. There's one question right now, but if you have other questions, then please put them in. You can also upvote other people's questions. And if we run out of questions on Whova, we can run around with the mic as well. But if we can get questions on here, that would be helpful. Thank you. We'll get to the questions because they're rolling in. So let's get started. So thank you for sharing about all the alternatives. What does an effective theory of change for policies on animal testing look like? What are the lowest hanging fruits on interventions?
Okay, I think I can take that. So the lowest hanging fruit on interventions has been cosmetic testing, and that's the reason that that has gained so much traction and everyone around the world are slowly working towards it. The next lowest hanging fruit would be the obsolete animal tests that already have alternatives available and or just require good manufacturing processes and nothing else in order to show that it's not toxic. So those are the lowest hanging fruits that are currently being worked on by different organizations. So the target animal batch safety test was one of those. So it just needs good manufacturing processes. So even though it did take quite a while because, as I said, regulators are very risk averse. So yeah, but that is something that we first tackle while also giving the opportunity to develop more alternatives in the space. And then you were talking about the theory of change?
Yeah, so there was one question about low-hanging fruits, and there was one question about what is an effective theory of change for changing policies on animal testing?
Effective theory of change. I think, so currently it's more on the lines of NGOs working in the sphere, finding out what the lowest hanging fruits are, taking proposals to the policy stakeholders, and then having them do something about it, and then speak to different industry stakeholders, and then get them all together. But I think a good theory of change would be a top-down approach, as I mentioned. So having better dialogue between different regulatory bodies. There are organizations that do that, like the OECD does conduct such conversations. But having more dialogue between policy stakeholders across borders, and then having a top-down approach where a policy is set in place, and then there's a trickle-down effect, and then there is more uptake or there is more development. That is something that would be most effective, in my opinion.
If I could also add to that, I think I come from an academic background, and I would say at this point, along with the top-down approach, it is also very important for the scientific community to get more involved and not just think about their research projects being something that they are passionate about and leave it over there. Most of the scientists, they develop non-animal methods or, as Brinda said, new approach methodologies, but it's just limited to the extent of how good their research is. And I think if they do not promote the research to such NGOs or communicate it to policy change makers or regulators, it is difficult to even adopt a theory of change, I would say.
Awesome, thank you. How much funding would it require to make a significant difference towards ending animal testing?
I can take that. Yes. I would say, in an ideal scenario, I would say all the fundings should be moved towards non-animal methodologies, but that's not practical at the moment.
All funding in the research.
In the research area. So like
Yeah.
I said, there were almost 14 billion US dollars that's being spent just on animal testing. So if you want a number, I would say that 14 billion should be slowly moved to researchers or research projects or organizations that are focusing not just on making their models more animal-free, but also who are trying to adopt to new approach methodologies. So that way you bring a larger group of people together. So, do you have anything to add?
Thank you. How to ensure the quality of vaccines without animal testing?
So one is good manufacturing processes. So when regulatory testing was first mandated somewhere in the 1930s, and in the 1930s there were no good manufacturing processes. So sterility was not it was difficult to sterilize the place, or it was difficult to effectively sterilize. It was difficult to see if the what do you say, not the supply chain, the manufacturing belts have effective sterility programs or sterilizing systems in place. I'm sorry. So it was difficult to monitor that. It was difficult to ensure that in the 1930s. But now we are in 2024, and technology has gone beyond leaps and bounds, and it's very easy to make sure that there are systems in place that ensure the quality control and quality assurance in a manufacturing unit. So good manufacturing process is one way of ensuring that you don't require certain animal tests in vaccine testing. So vaccine testing, is it vaccine testing that's written, or vaccine manufacturing?
Testing.
Vaccine testing. So there's also vaccine development and research. So that one, till now, they still use animals. But if you're only talking about marketing vaccines, which also requires a lot of animal testing, then good manufacturing processes to make sure that the vaccine is produced in a sterile way and is not going to be toxic when injected into humans. That is one way. And then there are also a lot of non-animal methodologies for testing safety of the vaccine that are out there, and which are in the process of being introduced to the regulators and introduced to uptake. So we need two things. We need good manufacturing processes, and we need more non-animal methodologies.
Can I add to that?
Of course.
I totally agree with Brinda. And another thing that I also keep wondering about is that the whole idea of trust, because we have been trusting animal testing for all this while, and it took a while to gain that trust, right? So I think it's also important to like we were talking about misguiding the public, right? So it's also important to show small achievements, like, you know, both in the case of vaccine testing, in the case of drug testing, chemicals testings, it's important to show the public that even a tiny reduction in the development of, say, drug X has helped to still promote or still market the drug. So the public knows that, okay, I can still slowly move away from thinking about whether my cosmetic or whether my drug has been tested on animals or not. And I think there's a growing population of people who always try to check if the drug has, or if the consumables that they are using are non-animal testing. So I think there are two sides to this problem, which is both from the policymakers, the regulators, the manufacturers, the scientists, and the academicians, and the public to work together towards this issue.
Thank you. How can we bridge the divides between policymakers, academics, and animal advocates?
Collaboration, getting them on the same platform. This is something that a lot of NGOs have been working on, where they are the ones who have dialogue with all three stakeholders. and then they organize certain workshops to bring them onto the same platform to discuss a particular topic. So this is how it has been conducted, or this is how a lot of change has been brought about. And another thing that is done is globally, there needs to be a lot of global dialogue that takes place. So if say the EU has banned something, then it's easier for India to look at the EU banning it, get more trust, and then implement that or move towards implementing or banning a certain test. So yeah, getting them on the same platform and having more dialogue and having more workshops, that is how you can bridge the gap.
Do you think that AI will play a role in ending animal testing, and if so, how?
Of course. Should we get? Yeah, so this is a very good question. So Dr. Peter Singer, his research associate Phi, a few others, and I are co-authoring a paper, and Dr. Thomas Harting are co-authoring a paper on using AI for good, which is using AI to end animal testing. So a lot of methodologies and a lot of computational methodologies, in fact, have AI inbuilt into them, and even predicting how a drug would work, or predicting which drug candidates would work well. This is all, it all has AI behind it, and of course AI does have a huge role to play in this. So this is one area that I would say that AI would play a really important role rather than something that we need to be scared of.
And I would just like to add that it would, when we were talking about resource intensive, it would drastically cut short the time that's being invested, because the in silico methods that Brinda was talking about, it usually uses these kind of models. And I think if you have AI used, or if you slowly move towards AI, I think it would be much easier to get a drug, to develop a drug and then get it approved to be sold in the market. So.
Thank you. Can we look at animal testing from a utilitarian angle and say, for example, that for vaccines, animal testing has more benefit for humans than does harm for animals?
Yeah, I would say no. because as we mentioned about translatability, a lot of these vaccines that probably work in animals, or a lot of the drugs that probably work in animals, may not work in humans. For example, aspirin. Aspirin was developed in the 1800s, and at that time there was no regulatory requirement for testing these drugs on animals. But if aspirin was developed now, and because there is regulatory testing requirement where you have to test the drug on the animal, see if it works, and if it doesn't, then it doesn't go into the market, we would never have aspirin. We would never have this important drug because it is toxic to mice, it is toxic to rhesus monkeys, etc. So that is one aspect of how a lot of drugs that probably that don't work in animals could probably work in humans, and a lot of drugs that work in animals probably have a lot of failure rates, because there is around 89% of drugs that pass animal testing have failed human trials. And that's why I wouldn't say that there's any utilitarian aspect to this, because if even from a scientific perspective, animal testing is extremely bad science, and the translatability to humans, as we said, we are not 70 kg rats, is extremely low.
I would also just add to that, coming from the scientific part of it, is normally when you do the safety assessment testing of different drugs and of different chemicals, you expose the animals, and it's something called the lethal concentration. That is where 50% of the animals die at that concentration. The issue is that none of us are exposed to that concentration. So that's the reason why we could still look at it, because I come from a middle ground here. We could still probably, I won't say we could look at it as a utilitarian aspect, because we know that several things has happened because we had animals as models, but several things has not happened just like, we wouldn't have aspirin because we have models. So yes, we can appreciate the fact that it has brought us to some level, but there are several crucial aspects that we need to pinpoint, such as the concentration levels that they are exposed to versus what we are exposed to.
Also chocolate. You wouldn't have chocolate today if it was tested on animals. So just putting it out there.
Cool. So what are the alternatives for psychological and neurological research that uses animals? And this person specified that they were talking about the, quote, meaningful ones, not the ones that were conducted with no real-life implications.
Should I take that? So I can shed some light on the neurological research, let's say. I know that at the moment there's a lot of research being done on brain-on-chips, where they try to understand Alzheimer's disease or the onset and progression of Alzheimer's disease. So I would say that there is research being done towards that direction. Are we there yet? Probably not. But again, through our presentation, that's what we were trying to say, is that we need more fundings so that more and more researchers, more and more neurologists or neuroscientists can move towards that direction. About the psychology, I don't know if you have something to say. One point that comes to my head is that, to my mind, is that most of these animals who are kept in cages, yes, there are a lot of regulatory guidelines on how they need to be treated, but from the background where I come, from the academic background of mine, and through my experience, I could say that most of the guidelines are not followed. So most of the mice or rats or even monkeys that are caged, they go through a lot of mental distress. And that is, again, something we wouldn't go through. But are there any developments yet in understanding the psychological aspects? I would say no, not yet.
Yeah, I think it also, because animal testing is still seen as the gold standard, and because it is so normalized and everyone considers that that is something that is required for human benefit, I think that that's also a major reason that's not pushing researchers beyond their boundaries and pushing them out of the box or to think outside the cage. And that's the reason why the limits are not being tested yet. As I mentioned with cosmetic testing, the ban was first proposed, and that's what pushed the industry to work more on non-animal methodologies before the ban was enforced. So we need more of this push in all of these areas where we think that there is nothing, because right now I don't think we are pushing ourselves to that limit, and there are a lot of barriers, as I mentioned, around researchers doing that.
And in following up with the previous question on AI, I know that researchers in the Johns Hopkins University, they are also trying to use AI to understand the neurological connections in the brain, and also to maybe, hopefully, way in the future, they would probably start understanding the psychology even for a particular mental health defect, let's say. So.
Thank you. So if you have any other questions, you can find Sreyoshee and Brinda at the conference at some point and ask them, and I'm sure they'll be able to answer any questions that you have. But we're going to wrap here. Thank you so much for coming, and have a wonderful rest of your day.
Thank you.